Hello and welcome back. We're going to finish up here with DNA replication. I talked a little bit about the replication fork, about the enzymes involved in DNA replication, the base pairing that happens. Now we're going to talk, and I know this is a little counterintuitive, but we're going to talk about how we start DNA replication, okay? Because DNA replication is going to start at specific sequences. And I'm not going to ask you to tell me what those specific sequences are, all right? But those sequences are known as an origin of replication, okay? In bacterial cells, there'll be one origin of replication for the entire DNA molecule that makes up bacteria. In eukaryotic cells, however, there are multiple origins of replication. And because of that, oh, that's squeaky. All right, what ends up happening is we get the formation of replication bubbles. Ooh, let me see. Maybe this will be better. That's a little better. Still squeaky. All right, so these weird bumps here, these are going to be my replication bubbles. Okay, and what we're going to have in our replication bubbles is we're going to have a particular sequence, and it'll be on both strands. That will be our origin of replication, all right? So that origin of replication is going to be where we start synthesizing our new strand of DNA, okay? So in this case, we're going to start synthesizing, boop, we'll go this way, oops, in our bubble, right? This one, we're going to start going this way, okay? That's what forms our bubble. And then what we have happen, that doesn't look very much like an arrow, let's fix that, okay, is the replication bubbles will grow closer and closer together until these strands of newly synthesized DNA meet, and then DNA ligase will come in and seal those up, okay? So what ends up happening, okay, is you're going to have that DNA replicate at multiple spots on the chromosome, all right? And I know we haven't talked about chromosomes yet, but we're getting there, all right? Those chromosomes, in the case of eukaryotic cells, is going to be a continuous long linear macromolecule of DNA, all right? So in terms of our replication bubble, we're going to have replication that's going to happen. We're going to go both directions, all right? And then eventually these meet up and they'll seal together to make one long continuous, oop, oh, terribly squeaky. New strand of DNA, okay? So our green, remember, this is our new strand. This is our parental strand, okay? And you can physically see these replication bubbles on chromosomes that are actively undergoing DNA replication using electron microscopy. Okay, so we can visualize, we know these replication bubbles happen. So I just wanted to talk a little bit about how things start, and that seems kind of weird to talk about how things start at the end, but I want you to understand the basic processes of what's going on here. You've got a replication bubble where DNA synthesis will happen, okay, because we're talking eukaryotic chromosomes, and it would be highly inefficient if we only started at one end and went all the way down the millions of nucleotides that make up a chromosome. So we've got multiple bubbles so that DNA can be replicated more quickly. All right. Hopefully that makes sense. If you have any questions, please let me know.